has to be copied before cell divide. This process is called DNA replication. This is a parental DNA molecule which is made of nucleotides joined into long strands by covalent bond. DNA's nucleotides are made up of 3 basic components: a 5-carbon sugar called deoxyribose, a phosphate group, and a nitrogenous base. DNA has 4 kinds of nitrogenous bases: adenine, thymine, guanine, cytosine. Thymine could pair with adenine. Guanine could pair with cytosine. The process of DNA replication begins with the replication bubble. In this bubble are 2 strands that will replicate the DNA. However, the original DNA must split for them to be duplicated. Because of this, in the replication bubble there are points where the DNA splits. These points are called replication forks. During DNA replication, the replication forks are responsible for splitting the connected strands of DNA. The DNA will then split itself into fork, becoming ready for the 2 strands responsible for replication. Helicase separates the 2 DNA strands by breaking the weak hydrogen bonds. DNA polymers is an enzyme that is most important in DNA replication, as it is the enzyme that goes through the replication bubble and creates new strands of DNA that connect to old ones. DNA polymers does this by joining individual nucleotides, and in the result, new strands of DNA are formed. The 2 strands of DNA molecules are leading strand and the lagging strand. The leading strand is called the leading strand simply because it leads ahead of the lagging strand. The leading strand originated from the origin of replication and continue along the split strand of DNA, connecting new nucleotides to form a new strand of DNA. However, the leading strand and lagging strand are active simultaneously. The lagging strand begins from its replication fork, or the area where the original DNA strand is split and divided into 2. As the leading strand is doing its job, the lagging strand goes through its route as well. The lagging strands go from the replication fork to the origin of the replication. Once it reaches its end, a new lagging strand will come in from where the first one started and continue doing its job until there is a replication strand of DNA. What these strands do are go through the replication bubble and fork all the way through the DNA until the original strand has been duplicated into 2 strands of DNA. The lagging strand leaves a few bases unpaired simply because they cannot connect them due to their sizes. These unpaired bases and pieces in daughter strand are known as Okizaki fragments. To complete the split of the DNA and its replication, the Okizaki fragments must be dealt with DNA ligase, go through the replication fork and bubble to each Okizaki fragment and connects them so that the DNA strand has been completely replicated. Once the Okizaki fragments have been paired by DNA ligase, the replication process is finished and the DNA strand has been replicated from 1 strand to 2 daughter strands.